We are now in the last part of the central dogma, and that is translation or protein synthesis. To recall, we have discussed how DNA is transcribed to mRNA so that the message can leave the nucleus without potentially damaging the DNA. Now we will discuss how the message in the DNA is translated into something tangible, as it can now be used to manufacture proteins. The message in the mRNA is coded in sets of 3 nucleotides. We call these the codons. Each codon, as you will see in the next slides, corresponds to an amino acid, which is the building block of proteins. There are a total of 64 different codons, and 2 to 4 of them correspond to one of the 20 types of amino acids. It is important to note that the order of codons determines the sequence of amino acids produced. The sequence of amino acids, on the other hand, determines what kind of protein is produced. A single difference in the amino acid sequence can create a different protein. So how do we know where to start? The first thing that you have to remember is that translation always begins with the codon AUG. This is known as the start codon. Once you find AUG, you can then divide the rest of the nucleotides into clusters of 3. AUG, or the start codon, establishes what is called the reading frame. How do you know when to end? Translation ends when you encounter any of the following codons: UAA, UAG, or UGA, UA, UAG, or UGA. These 3 are known as the stop codons. Once you have successfully established the reading frame, you can use a codon table, like what you see on the screen, to determine what amino acid the codon corresponds to. The left column lists the codon's first base or letter. The top row lists the second, and the right column lists the third. 61 of the triplet codes for amino acids. One of those, AUG, both codes for methionine and serves as a signal to start translation. 3 codons are signals that stop translation. So what is the amino acid sequence of the mRNA on the slide? Pause the video to try it out. The resulting amino acid sequence is methionine or met, followed by leucine or leu, then glycine or GLN, then alanine or ALA, and after that would be the stop codon UGA. Hopefully, you now understand how translation generally works. But what actually happens in our cell during translation? There are 3 RNA types that are used in the translation process. Number 1, your messenger RNA. Your messenger RNA produced during transcription carries the message encoded in the DNA. Number 2 is your ribosomal RNA or rRNA that comprises the organelles called ribosomes, where the process of translation occurs. And lastly, number 3, you have the transfer RNA or tRNA that delivers the amino acids matching the mRNA codons to the ribosomes. The real MVP of this process is the tRNA. Take a look at its structure on the slide. On one end, it has a sequence which is called the anticodon. The anticodon matches with the codons in the mRNA. What do you think is the codon that matches with the anticodon ACC? Yes, it is UGG. On the other end of the tRNA is the amino acid sequence that actually corresponds to the codon UGG, which is tryptophan or TRP. Now let's go to the actual process of translation. It involves a cellular organelle called the ribosome. The ribosome has two parts, a small subunit where the mRNA attaches to, and a large subunit where the tRNA molecules enter. The large subunit has three chambers. The first chamber is called the amino acyl tRNA binding site, but let's just call it A site, which basically is the entrance of the tRNA to the ribosome. This is followed by the peptidyl tRNA binding site, or the P site, where peptide bonds are created between amino acids. Lastly, you have the exit site, or E site, where used tRNA exits. Let's backtrack a bit. Do you remember what peptide bonds are? Peptide bonds are covalent bonds between two amino acids, which, upon reaching a particular length, becomes a polypeptide and eventually a functional protein. During translation, the mRNA attaches to the small ribosomal subunit. Then the large subunit attaches to it. Now complete, the ribosome then scans the entire mRNA strand until it finds the start codon, AUG. The ribosome can now establish the reading frame. Once it has locked AUG in the A site, it then attracts a tRNA that has the anticodon for AUG, which is UAC. You can see that the tRNA carries on one end the amino acid methionine or MET. The ribosome then moves to the next codon, which is GUG, and recruits a tRNA with the anticodon for it, which is CAC. This tRNA carries with it the amino acid valine or VAL. The tRNA carrying the methionine amino acid then moves to the P site as the new tRNA carrying valine occupies the A site. In the P site, a peptide bond is formed between the two amino acids, effectively connecting them. The ribosome reads the next codon, UUA, and again recruits the next tRNA with anticodon AAU. This tRNA carries the amino acid leucine or LEU. Since there is a third tRNA, the first and second tRNAs move to the E and P sites respectively. When the first tRNA arrives at the E or exit site, it is released out of the ribosome. But since the amino acid methionine it was carrying was already bonded to valine, it remains attached to the growing polypeptide chain. Now that valine is in the P site, it forms a peptide bond with leucine. This process continues until the ribosome encounters a stop codon. If it does encounter a stop codon, the chained amino acids are released out of the ribosome. The amino acid chain, now called a polypeptide chain, is folded to take on a 3D shape. This is now a functional protein that the cell can use. As a review of translation, in eukaryotes, RNAs are transcribed in the nucleus, then transported into the cytoplasm. Translation begins when ribosomal subunits and tRNA converge on an mRNA in the cytoplasm. Then, tRNAs deliver amino acids in the order dictated by successive codons in the mRNA. The ribosome links the amino acids together as it moves along the mRNA, so a polypeptide forms and elongates. But what does happen if there are mutations in the DNA? Mutations happen when there are errors in the letter sequence in the DNA. Mutations can go three ways. Number one, they can have no effect and you go on with your life not knowing that you're already a mutant. Number two, they can change the protein product into something that's beneficial to you. And number three, they can change the protein product into something that's lethal, which almost always causes the mutant to have complications or die. We will be discussing three types of DNA mutations: missense mutations, nonsense mutations, and frame shift mutations. The first type is a missense mutation, otherwise known as base pair substitution mutations. This mutation occurs when one letter or one nucleotide from the original DNA sequence is replaced with another letter. In the example, you can see that thymine, or T, was replaced with adenine, or A. Because of this base pair substitution, the resulting mRNA is different. Instead of adenine, uracil is transcribed. Because of this change in the mRNA sequence, the resulting amino acid is also changed. From glutamate, it becomes valine. Usually, base pair substitutions don't lead to lethal conditions. Unfortunately, in this example, this single nucleotide or letter change leads to a condition known as sickle cell anemia, in which red blood cells become crescent-shaped. This mutation is common in many African countries. The second type of mutation is called a nonsense mutation. It is a type of base pair substitution in which the resulting mRNA accidentally forms a stop codon. In the example, let's say this is your DNA sequence. The resulting mRNA sequence would be the following, which will lead to this amino acid sequence. However, if the DNA sequence has a base pair substitution, in which A is replaced with a T, the resulting mRNA will also change from a U to an A. The new mRNA sequence will be as follows. You'll notice that instead of UUA, which corresponds to the amino acid leucine, you have UAA, which is a stop codon. So how will this affect the protein? Instead of having a long protein chain, it is prematurely terminated because of the creation of the stop codon. The protein will ultimately become dysfunctional and cannot be used by the cell. One of the most dangerous mutations is what you call the frame shift mutations, because these can shift the reading frame during translation. This is caused by either the deletion or insertion of one or two bases. To illustrate this easier, let's say you have the following sentence composed of three-letter words. The fat cat ate the rat. It can be read easily, right? Now what if I delete the letter F and ask you to read it in three-word intervals? The at, ata, tet, her, at. Does it make sense? Now, what if instead of deleting a letter, I add the letter C and again ask you to read it in three-word intervals? The kfa, tka, tat, era, ti. Again, doesn't make sense. Note that because the entire reading frame is shifted, you can potentially change the entire amino acid sequence, and with that, the protein product.